This program is made possible by a grant from the National Science Foundation, a co-production by Mississippi Public Broadcasting and the University of Southern Mississippi Gulf Coast Research Laboratory. April 20th, 2010, 41 miles offshore in the Gulf of Mexico, BP's Deepwater Horizon drilling unit exploded after high-pressure methane gas engulfed the rig and exploded. 11 people would lose their lives that night. For nearly 3 months, oil gushed into the Gulf of Mexico. Marine life, tourism, and petroleum operations will never be the same in the Gulf region. Now there are questions that have scientists focusing on trying to shed light on the unknown long-term effects on the Gulf. This is the Science of the Spill.
Hello and welcome to the Science of the Spill. I'm Erin Pickens.
And I'm Dr. Bob Thomas, Director of the Center for Environmental Communication at Loyola University.
Well, the Science of the Spill is a three-part series produced by the University of Southern Mississippi Gulf Coast Research Laboratory and the Mississippi Public Broadcasting to inform the public with scientifically proven facts concerning the BP Deepwater Horizon oil spill. And Dr. Thomas, I think we can all agree that there are many myths and information out there that are not based on scientific data about the effects of the spill.
Well, that's right, Erin. And we're going to bring together scientists that are studying this disaster for discussion to give the facts about issues related to the Deepwater Horizon oil spill. In today's program, we'll be discussing the impact of dispersants on the environment and also the overall health of the marine life in the Gulf.
Now, we will also let you know that you can find more on our companion website at spillscience.com. There you can find in-depth information about topics related to the spill, as well as video discussions from scientists studying the effects of the spill on the Gulf of Mexico. You may also want to join the discussion on our blog section of the site, where you can engage in discussions with scientists studying the impacts from the Deepwater Horizon oil spill. Again, that's spillscience.com. Now, during the early stages of the spill, a large amount of dispersants were dropped to help break up oil found on the top of the water. Southern Miss scientists at the Gulf Coast Research Lab and CINIS have posed many questions about the impacts of dispersant agents often used to respond to oil spills.
The dispersant issue is a very interesting one. The idea of putting the dispersants in with the oil is in principle a good idea. You'd like to widely disperse the oil to make it more available for microbial degradation. That's the basic concept. Now, the problem, of course, is that you're adding another chemical to the mix.
The main dispersant that has been used is a compound made by a company, Nalco, and it's their Corexit 9500. There are 6 ingredients in Corexit 9500s. All 6 of them have been approved for various uses in human products, ranging from toothpaste to pharmaceuticals to propylene glycol as antifreeze in your radiator. And propylene glycol is probably one of the most toxic compounds, and it's not terribly toxic. So the role of these dispersants is to work as a surfactive agent. And a surfactive agent is nothing more than a detergent. One end of the molecule likes oil, the other end of the molecule likes water, and so it physically pulls the oil apart into very small pieces that the microbes can then interact with.
One thing that's kind of interesting in those numbers that I really haven't seen much public comment on is they say that 8% of the oil was chemically dispersed. Now, of course, these dispersants are things that people have been very concerned about. So a question to be asked is, first of all, how good a number is that 8%? And if that's it, then from a policy perspective, does it make sense then to use the dispersants if that's all that it's affecting?
The controversy is that some scientists believe that the dispersant is toxic and that we're underestimating the toxic effects. Other scientists believe that it's not terribly toxic, but like all chemicals, if you apply enough of it, it can be toxic in large quantities.
EPA has a certain set of tests that it runs mainly on one type of organism looking at toxicity. Some people have suggested that maybe there need to be toxicological studies on different sorts of organisms and perhaps for longer periods of time. And in fact, some of the research that the National Science Foundation has funded for this oil spill is indeed looking at some of those questions.
The one thing that I have done in my laboratory is to look at the decomposition of Corexit. And in my laboratory, in what we call microcosms, simulated replicas of the environment, we've simply come down here to this dock, collected water from this bayou, and we know full well that there are oil-degrading microbes in this water, and we've used that to take a look in my lab at the decomposition of oil as well as the decomposition of Corexit. As scientists look at the fate and effect of dispersant in salt marshes, in marine animals, we'll have a much better handle on what the effect of the dispersant is as a result of the Deepwater Horizon incident than we did before.
Now, we've heard some important questions raised by Dr. Schiller and Dr. Grimes about the use and effects of dispersants. We have Dr. Thomas now here with scientists that can help answer some of those questions. Dr. Thomas.
Well, thank you, Aaron. I'm here today with Dr. Joe Griffith, who's an assistant professor in the Department of Coastal Studies at the Gulf Coast Research Lab, and Dr. Christine Willett, who is a professor in pharmacology and environmental toxicology at the University of Mississippi. Welcome. Good to have you with us. If we could start today by talking a little bit about your research program, and then we'll get into some specifics about how that affects the work on the Gulf. Okay. Christine?
So I study the mechanisms that a particular class of chemicals that's in oil, the polycyclic aromatic hydrocarbons, and we typically just call them PAHs. And I study how they cause their toxicity. And so we've been using primarily fish models, and we like to look at these chemicals are known carcinogens, and so we study about how they cause cancer, but then we're also moving now into how they might cause this thing called endocrine disruption, how they might interfere with hormones, and also maybe reproductive effects. And so as we move into the oil, then we're thinking about whether the oil could potentially be causing some of these same things. In addition to that, we have an analytical aspect of our research, wherein we just collect samples along the coast, like water and sediment, for example, and measure the concentrations of PAHs in those. We actually started this project right after Hurricane Katrina hit, so we sort of saw the impact of Hurricane Katrina, and now those levels have returned to baseline, and so now we're poised to see the impact of the Deepwater Horizon spill at those same sites. And those sites were from Gulfport to Mobile, so we have a nice chunk of the Mississippi coastline to review.
Well, it's comforting to know that you have a baseline of information you can go back and compare it to now. Joe?
Yeah, so as soon as the incident happened, my lab started focusing on two different lines of research. One, we went on the boat 3 or 4 days after the Deepwater Horizon rig collapsed, and we were sampling in the Mississippi Sound and along the coastline of southern Mississippi for species to establish our own baseline. So we collected a large number of samples. I think the last time I checked, we've got 970 samples sitting in the freezer of different species, finfish, shellfish, oysters, things like this, so that we would be able to go back to the same sites later on and establish exactly what the effect was by examining the differences between the two. So we've got a large number of species in the freezer ready to be analyzed. Simultaneously with that, we're doing some laboratory-based studies. We've obtained crude oil and dispersant from BP, and we're mixing that together to produce what we call dispersed oil, essentially the mixture of oil and Corexit that we hypothesize the animals are being exposed to in the Gulf. And we're exposing different species to that under very carefully controlled laboratory conditions. The idea being that in the laboratory we can isolate out all the different environmental effects that might be confounding the response and isolate and identify just exactly what the effect of the dispersed oil on different organisms is. And we have 3 different exposure chambers in the toxicology building at the Gulf Coast Research Lab. We've got all 3 of them busy constantly. We have one that just started on Friday. We're looking at chronic effects of dispersed oil exposure to a small fish that's indigenous to the region called the sheepshead minnow. We're simultaneously running exposures to some of the shrimp species that are indigenous to the area. We grow those in culture at the Gulf Coast Research Lab, and so we're doing exposures both on adults and juvenile examples of those species. And we have, along with my colleagues, a NSF Rapid grant to look at the effects on blue crab larvae. So we're exposing those guys to crude and dispersed oil to identify the effects on them as well.
Well, that's excellent. From the video, I think it was pretty apparent that there are some bona fide reasons for using dispersants when you have an oil spill. I'd like each of you to comment on it in your labs. What are you seeing about the residual toxicity of the dispersants and the breakdown components?
Well, I'm really not going to talk too much about what we've seen. It's still very, very preliminary, and we want to make sure that we have it vetted through peer review to make sure that our results are valid and that they're agreed with. We're much more concerned with being accurate than we are with being right on this issue. I'm sorry, we're more concerned with being accurate than with being quick on this issue. Mm-hmm. I understand that people want to understand, want to know what's going on, but nobody's served if we have information in the press or in the public that later turns out to be inaccurate. So we're going to wait until we have our results reviewed by other scientists, and then we'll release it.
Good. Well, would you, and you kind of answered my next question right there at the end, but before we go to Dr. Willett, tell the audience what you mean by peer review and why that's so important to a scientist.
Sure. So peer review is essentially the way that we try our best to make sure that no inaccurate information gets published. And it's not perfect, no process ever is. But essentially we'll complete a section of research, we'll come to a conclusion about the effects, and we'll write it in a paper for publication. And we'll send it to a journal, and the journal will then send it out to other scientists entirely unconnected to this. I don't know who the reviewers are, and they'll read through the paper very, very carefully, and then they'll try to tear it apart. They'll try to find every weak point in it, and then I'll get a version of it back in the journal. I say, well, it's rejected or it's accepted, but you have to address these comments. And it's a series of very rigorous, very focused comments on exactly what is this, why did you do this, how do you explain this in light of this, and then I have to write a response to that addresses each of those concerns, so that by the time it's been out and it's actually published, it's been through one, maybe two rounds of review where other people who aren't invested in this research are making sure that what I'm saying is accurate.
Well said.
Sure.
Dr. Willett.
Yes. So the question is back to what the dispersants. So we haven't been addressing that exactly, but what I can say is that the potential from the dispersants is now that the chemicals that are in the oil are now dispersed through the water column. So if you've got the surface slick on the top, then organisms that have to come up through that slick, your sea turtles, your dolphins, and things are getting exposed to that slick. But potentially things under that may or may not have an impact. Well, once you disperse that oil, things like seagrass habitats or oyster reefs or so forth could now be, those oil constituents could now be bioavailable. So some of the work we're doing is we've collected, right as the spill happened, we collected seagrasses and oysters, and we've been collecting those throughout this time course. And what we're going to try and do is see so those ones before the spill are our sort of baseline, as you might say. But then we predict there might be some sort of stress response in those organisms to oil or dispersant or whatever was there. And then we're going to look for a return to that original condition or recovery. And we're doing this by what's called proteomics. And so we're looking for a suite of different proteins that are expressed in those organisms to see if we can, sort of track this change, track the stress, and then perhaps the recovery. So again, these organisms might not have been exposed if the oil had stayed on the surface, but potentially now we might have impacts in those kinds of organisms that live, below the water line.
Okay. Now